In this video, we will discuss about cell wall, types of cell wall, structure of cell wall, and function of cell wall. As you know that cell wall for the first time discovered by a scientist named is Robert Hooke in 1665 while he studying thin slices of cork of the quercus plants. Basically, he discovered dead cell. Within the dead cell, he discovered the cell wall. In 1804, Carl Rodolphi and G.H.F. Link is the name of the scientist who proved that cell wall have independent cell wall. Cell wall can be defined outermost covering in plant, bacteria, and fungus is known as cell wall. It is basically cell wall produce formed boundary of the cells. On the basis of chemical composition of cell wall, there are three types of cell wall. Bacterial cell wall, which is made up of peptidoglycan or murine. Fungal cell wall, which is made up of chitin. And plant cell wall, which is made up of cellulose. Bacteria. The science of bacteria is known as bacteriology, which is a branch of microbiology. And bacteria for the first time discovered by Anton van Leeuwenhoek in 1670. In this diagram, you can see the bacteria can be divided in response to stain into two types, gram-positive bacteria and gram-negative bacteria. Gram-positive bacteria have thick peptidoglycan as indicated here in green color, and gram-negative bacteria as indicated in the picture a thin green color line. Along with the peptidoglycan in gram-negative bacteria, outer membrane of lipopolysaccharides and periplasmic space is also present. That's why we can say that the cell walls in gram-negative bacteria is more complex and the cell wall of gram-negative bacteria are more resistant than gram-positive bacteria because the outer layer of lipopolysaccharide prevent the entry of antibiotics. So we can say that gram-negative bacteria are more pathogenic and it show more resistivity to the antibiotics. So they are more pathogenic as compared to the gram-positive bacteria. Peptidoglycan is it is commonly known as a murine. It is a carbohydrate protein complex. It is a carbohydrate protein complex. In the diagram, you can see that the nitrogen acetyl muramic acid is a peptide chain of 3 to 5 amino acid. The peptide chain can be cross-linked to the peptide chain of in another strand forming the peptidoglycan. These are the structure of peptidoglycan which form the cell wall of gram-positive and gram-negative bacteria. As I have told that Hans Christian Gram, a Danish physician, he in 1884 divided the bacteria into two types, gram-positive bacteria and gram-negative bacteria on response to stain, on the basis of stain. So gram-positive bacteria are those bacteria which are stained blue-purple with crystal violet dye, possesses thick walls of peptidoglycan. They retain the dye when the cells are washed with an organic solvent like alcohols. While gram-negative bacteria, they does not retain the dyes. As I have told that gram-positive bacteria, their cell wall is usually thick and mainly composed of peptidoglycan, 50 to 90% of the dry weight of the cell wall, and contain large amount of teichoic acid. And as I have told that the cell wall of gram-negative bacteria is more complex. The thin peptidoglycan layer is covered externally by a layer of lipopolysaccharides. And this lipopolysaccharides, along with this complex structure, it gave resistivity to various types of antibiotics. In this diagram, you can see that Hans Christian Gram is the name of scientist who divided the bacteria into gram-positive and gram-negative on response to staining procedures. Function of the bacterial cell wall, as I have told that it gave proper shape to the bacterial cell. It prevent various types of material into the bacteria and from bacteria to the environment, and it control the transport of various types of material, and it prevent various types of harm and injuries to the bacteria. It also take part in protein synthesis. It also control the movement of cells, and it also control minerals within the cell wall. Fungi are achloropilus, heterotrophic, and multicellular organism, and the science of fungi is known as mycology. The fungal cell wall, as I have told, it is made up of chitin. It is made up of chitin. And chitin basically polymer of acetylated amino sugar N nitrogen, acetyl glucosamine, glucans and protein also form the chitin. In this diagram, you can see the structure of the fungal cell wall. Function of a fungal cell wall: it protects against osmotic lysis, it gives proper shape to the fungal cell, it control the transport of various types of material into the fungal cell and outside the fungal cell, having binding sites for enzymes. The plant cell wall, as I told that plant cell wall was discovered by Robert Hooke in 1665, earlier than the protoplast. And basically, the plant cell wall composed of the middle lamella, the primary cell wall, the secondary cell wall, and the tertiary cell wall. In this diagram, you can see the middle lamella. It is the outermost structure, or it is the structure which is present between the two cells, and it acts like a cement between the two bricks. So this is known as middle lamella. This bluish color, it is known as primary cell wall, while yellowish color, it is the plasma membrane in this diagram. Primary middle lamella, first of all, their thickness is 1 micron meter in thickness. Their thickness is 1 micron meter in thickness. It is present between two adjacent cells. It is situated outside the primary cell wall, and it is made up of calcium and magnesium pectate. It is a cement which holds the adjacent cell together. In this diagram, you see the black line represent the middle lamella, primary cell wall. The thickness of the primary cell wall varies within the plant cell. It is usually 1 to 3 micron meter in thickness. It is more elastic and extendable, crystalline and optically active. It is formed after the middle lamella, a thin, flexible and extensible layer. It is capable of growth and expansion. It is the backbone of the primary cell wall, made up of cellulose fibrils. The matrix is composed of hemicellulose, pectin compounds, lipids, structural protein. You can see here the primary cell wall composition: hemicellulose, pectin, cellulose, microfibril, and sometimes soluble protein is also present within the primary cell wall, while green color it here represents middle lamella. The secondary cell wall, as I have told that primary cell wall is present in all plant cells, while secondary cell wall is present in those cells which have differentiated. Primary cell, it is extremely rigid and provide strength. And the thickness of the secondary cell wall is from 5 to 10 micron meter. It is not, as I have told, it is not found in all cell types. It consists of three layers known as S1. Here in the pictures or in the diagram, this structure is known as the S1 layer, S2 middle, and S3 inner. So this is the innermost layer, it is the middle layer, and it is the outermost layer. And the secondary cell wall chemically composed of cellulose, hemicellulose, and lignin. And tertiary cell wall is present in those plant cells which have much thick cell wall. Tertiary cell wall is deposited in few cells. It is considered to be dry residue of a protoplast. Beside cellulose and hemicellulose, xylans is also present. And plasmodesmata can be defined. It is a cytoplasmic connection between the two cells. A cytoplasmic connection between the two cells are plasmodesmata are protoplasmic strand that connect the protoplast of neighboring cell, and the diameter of plasmodesmata is 40 to 50 micron meter. Here the cell wall is basically absent where plasmodesmata is present. Chemically, as I have told, that cell wall is basically composed of cellulose, which is a type of polysaccharides, pectins, hemicellulose, xylans, and as well as the protein. In this table, you can see the differences between the primary cell wall and secondary cell wall. Various types of features are present there: occurrence, position, nature, etc., etc. These are the differences. The function of cell wall. The first function of the cell wall of the plant, it gives proper shape to the plant cell. It gives proper shape to the plant cell. That's why it determines the morphology, growth, and development. And they protect the protoplasm from invasion by viral, bacterial, and fungal pathogen, or from any other physical type of injury. They are rigid structure, thus they help in plant in withstanding the gravitational forces. It also controls the transport of various types of material into the plant cell and outside the plant cell. They withstand the turgor pressure which develop within the cell due to high osmotic pressures. And there are some other uses of the cell wall of the plant cell, and they are used industrially. Various types of industry Totally depends upon the cell wall. For example, an explosive material, nitrocellulose, is obtained from cell wall. Textile fiber, rayon, is also a type of cell wall. Cellophane, which is a partially permeable membrane, is also a type of cell wall. And plastics, including celluloids and cinematography, is also a type of cell wall. And cell wall is also used in paper making. Is also used in paper making. If you like the video, please share, subscribe, and watch.